The experiment for today is about separation of amino acids by thin layer chromatography. The principle behind the experiment is the separation can be done based on the distribution among two phases, namely the stationary phase and the mobile phase. The stationary phase used in this experiment is the thin layer of silica applied on the glass or aluminum support. The mobile phase is the solvent mixture used in this experiment. The separation depends on various factors, which are the solubility of the compound and the attraction between the silica and the substance to be separated. If the amino acid that interacts strongly with the silica moves to lesser distance, and if the amino acid that weakly associated with the silica moves to the greater distance. An important factor in TLC is the RF factor. RF is the distance moved by the solute from the origin to the distance moved by the solvent from the origin. And amino acids which are separated in the TLC are found to be colorless. You can't see through the naked eye, and which can be identified using the locating agent called the ninhydrin. Ninhydrin reacts with alpha amino acid and results in a purple color spot, which you call it as Rohmann's purple. The materials required for the experiments were standard amino acids and unknown, distilled water, pre-coated TLC plate, TLC chamber, butanol and acetic acid, spraying reagent ninhydrin, hot air oven, scissors, pencil and ruler. To begin with the procedure for TLC, the TLC plate was taken using the tissue paper and forceps, because the hand sweat containing the amino acid will contaminate the TLC plate. The TLC plate of desired length was cut using the scissor and leaving 0.5 centimeter from the bottom, draw a line, a thin line, using the pencil and the ruler. Now mark the spot, leaving sufficient distance to load your standard amino acids and unknown. Load the amino acid using the capillary tube. Focus. Load the standard amino acids and the unknown in the respective spot marked on the TLC plate using the capillary tube. Once the sample is loaded on the plate, leave the plate for air drying. Meantime, you can prepare the TLC chamber by preparing the solvent mixture of butanol, acetic acid and water in the ratio of 4:1:1 for around 5 ml, and pour it in the chamber and leave the chamber undisturbed for saturation. Once the chamber is saturated for 30 minutes, now you can take the TLC plate using the forceps and place the plate inside the chamber for separation of amino acids. While placing the plate in the chamber, make sure the solvent should not be above the line drawn for loading the samples. because this will make your sample solubilized in the solvent mixture. The solvent will be taken in the paper by capillary action and leave the chamber undisturbed for the amino acid to separate. The cover of the chamber is open to take the paper out and check for the run of amino acids. You could find the mobile phase was moved up the plate by capillary action, carried the samples along with it. But it is impossible for you to see the amino acid through your naked eye. Now, the TLC plate is allowed to dry in hot air oven or using the hot plate at a temperature of 60 degree. So this makes your paper dry. Now the TLC plate is sprayed with the ninhydrin, the locating agent, to check for the Rohmann's purple spot to develop. After spraying the ninhydrin reagent, the TLC plate can be kept in the hot air oven or in the hot plate and make the plate dry and check for the development of the color or the spot in the paper. After development of the spot in the TLC plate, now it's the time to measure the RF value. RF, as I told you earlier, RF is the distance moved by the solute to the distance moved by the solvent from the origin. So calculate the distance moved by the solvent from the origin. Similarly, calculate the distance moved by each and every amino acids, including the unknown. Compare the RF of the unknown with the standard amino acid. So the RF which is matching with the standard amino acid will be your unknown. So unknown can be a mixture of amino acids or it can be a single amino acid.